So I'm going to cover a lot of territory in B cells, and I appreciate the introduction that Peter Lipsky provided, so I can glance over some of the material and just talk about it from my perspective. So we'll talk about therapeutic agents, and I thought that since I'd contract some of the earlier material, I might have some time to talk about some data from our own laboratory that have to do with autoantibodies that are distinctive and characteristic and maybe driving rheumatoid arthritis at the end. So let's just remember that every day of our lives, we continue to develop and differentiate new B lymphocytes. So we have the capacity to regenerate our repertoires, starting from precursors and stem cells in the bone marrow. There's sequential stages of differentiation and changes in cell surface molecules and signaling pathways to develop transitional B cells that are the earliest B cells with antigen receptors on their surface in the bloodstream, and going on to become mature B cells, differentiating to follicular B cells, marginal zone B cells that have distinct roles and respond to distinct antigens. And then if a B cell has the right specificity, it may be drawn into a germinal center response, activated, and the result would be either differentiation to a antibody-producing factory, short-lived plasmablast, or long-lived plasma cell that's found a niche and a home, presumably in the bone marrow and health, but maybe involved in niches created in inflammatory tissues during disease. And then you bank away memory cells. Memory is supposed to be they're antigen specific, and they're supposed to be more easily triggered by exposure to antigen, presumably so that past immune exposures to infectious agents will elicit more rapid immune responses in the future. But the problem is if you develop an autoimmune disease, you may well develop the memory of something bad. And that may be very difficult to actually cure an autoimmune disease because you have this banked memory, which we'll come back to. But with regard to the roles of B cells in health and disease, there are many roles. We talk about B cells actually producing LT beta, which is a chemoattractant. Chemokine helps attract inflammatory cells where there may be the development of secondary lymphoid tissue, and this has been shown to be relevant to the pathogenesis of several autoimmune diseases, as I indicate here. But very important is the role of B cells as antigen-presenting cells, and particularly after you've initiated your immune response. Maybe a dendritic cell is easier for eliciting immune recognition of a foreign antigen or even a self-antigen at the initiation of the disease. But after that, memory B cells may be the most efficient at taking up, processing, and presenting an antigen to help maintain and stimulate antigen-specific T cells. And the reason that I mention that, of course, is just to suggest to Eric that abatacept may not be a T cell targeted agent. And in fact, Artie Cavanaugh, wherever you are, may correct me, but as I recall, at UCSD they performed biopsy studies on rheumatoid synovium that were followed up after a course of abatacept, and they looked at the changes in the synovium. And what were they're right there, that's great. And what were the, so you can say I'm right or I'm wrong, but the most significant and dramatic changes detectable in rheumatoid synovium after treatment with abatacept was actually dropout of B cells. And so abatacept may be a B cell targeted agent, just getting rid of, and the idea is that the signals go from the antigen presenting cell to the T cell, and then the T cell through the same circuit actually provides stimulation for the antigen presenting cell, which, based on the immunohistologic data, suggests it's a B cell. So it's good to know that there are several classes of B cell targeted agents that have been embraced by the market. B cells can also be potent cytokine-producing cells, and I often get pushback where they say, but Greg, in rheumatoid synovium, there's much higher levels of production of TNF or IL-6 or other agents, often by a macrophage. But the point is the macrophage may be thought of as just sort of an automaton for integration of signals coming to it, but the B cell may be upstream, and there's also a concept of the pairing of an antigen-specific B cell and a T cell that provides what the other needs. They're the perfect couple, and you can have polarization for BE1 cells that help TH1 polarized cells, as well as TH2 biased cells. So they each provide cytokines that stimulates the other, and they help get each other through the immunologic morass. Now, autoantibodies, of course, are produced by B cells, and I just want to comment that only in a minority of diseases is actually the binding interaction of itself pathogenic and contribute to the disease. In most cases that we study, at least in rheumatology, it's immune complexes that form, and by forming these immune complexes, they gain the capacity to trigger complement and inflammation or engage in the activation of Fc gamma receptors. So implicit to all this, the pathogenesis of our diseases. So how do we target B cells? Well, sometimes very well, but there are different approaches. Of course, the old school prototype is this very decrepit now old school molecule that even has a little bit of mouse variable regions in it, but it's working great. It's like a Timex. Takes the ticket and keeps on whatever. But so Rituxan has was originally developed in San Diego. I have to say Mitch Reff, who made it with his two hands, as I like to say, used to go to the same synagogue I did. He's a wonderful person. And the trials were designed in San Diego, and fellows that I had in training actually led the pivotal trials in non-Hodgkin's lymphoma, as well as in rheumatoid arthritis. It actually only targets B cells at a certain stage of differentiation and does not affect stem cells, and has little or no effects on plasma cells. And the effects on memory are interesting, which we'll discuss in a moment. And this is approved for the treatment of TNF inadequate responders that have, with RA, and actually for the treatment of ANCA-positive vasculitis more recently. Belimumab, the prototype of a BAFF blocking antibody, interferes with the survival factors that Peter discussed, and there are a number of others that are in the pipeline, which we'll mention briefly. But let's just talk about the mainstay of B-cell targeted therapy, and CD20 is a very complicated transmembrane protein. It actually is now known to not only play roles in B-cell development, but to actually have signaling function, where there are certain tyrosine kinases that are triggered by interaction with CD20. The reality is that the development of this agent therapeutically, I believe, was one of practicality and convenience. Their trials with anti-idiotypic therapy at IDEC had shown efficacy, but you'd need a range of anti-idiotypes to target a large number of lymphomas, which were impractical to bring to the marketplace. So it is my impression, or my guess, or my fantasy, that the people running the company got together in a room and they said, well, if anti-idiotypes weren't practical, how could we develop an agent that targeted CD20 positive non-Hodgkin's lymphoma? And the other person said, I don't know. How do we target CD20 positive non-Hodgkin's lymphoma? So they decided to make an antibody to CD20, even though no one knew what CD20 did, and there were no mouse models, and there were actually no studies done with toxicity, and the entire pivotal trial was successful with a total of 161 human beings in it. And there you go. The rest of it is sort of history. And that's mostly true. So the first generation anti-CD20 agent has shown wonderful efficacy in a number of diseases and an attractive side effect profile. There used to be only 3 mechanisms by which it was thought to kill B cells, either through complement-dependent cytotoxicity, antibody-dependent cellular cytotoxicity, and actually by cross-linking the CD20 into lipid rafts, causing apoptosis. But now somebody's found a 4th mechanism to make it more complicated by induction of reactive oxygen species. So that may or may not be important, but what I want to highlight is the effects on different B-cell subsets, and Peter did a great job of explaining what otherwise might seem complicated, that there are different B-cell subsets and their roles functionally are very different. And by flow cytometry, you can actually just gate first on B cells by CD19, and then a lot of work has been done to try to figure out the effects on memory, because that's a very important component of the immune system, and with regard to whether you can actually long-term suppress or potentially one day cure the disease, and the idea that IgD is present on the most early and naive B cells, and CD20 is a relatively good marker of memory, and so there's actually IgD positive memory and IgD negative memory, which is called switch memory, which we'll get back to. But one of the most interesting things that Rituximab does, actually the most interesting thing I think, is that you can get an infusion of 2 grams, and that can result in no measurable blood B cells, and it doesn't really seem to matter. It's quite disappointing to a B-cell immunologist to find that there are no B cells in the bloodstream and people aren't really immunosuppressed, and they hardly know it at all, in fact. But the infusions are conducted, and you'll have no measurable B cells, and then slowly they'll come back. And the patients who have the best clinical responses have a shift in the subsets in their blood B cells. And the best responses are those, in fact, that have reductions in the previous representation of memory B cells, and what returns preferentially are naive B cells. So not everybody does this, but those that have the best clinical responses have sort of a reduction in the memory with at least representation. Now, we don't know if this is good memory or bad memory that's lost, but it is an interesting observation, and there was a fantastic paper that was in JX Med that suggested that this is a loss not only of memory B cells, but actually of IL-6 producing B cells, and IL-6 producing B cells may be uniquely able to actually recruit Th17 cells. So the B cells are pulling other people's strings, and a B-cell immunologist loves to hear that. All right, so we went through this very quickly. The point is, how does anti-CD20 therapy work? It kills some B cells, but how does it really work? And one hypothesis is that it cleans out nests of memory B cells and differentiation products at sites of disease because you have arthritis in your joints. So this is something that actually I think we should remind ourselves and understand what the agent does. We have to get past the blood and look in the affected joint. So let me just switch gears a little bit to talk about BLYS and APRIL, B lymphocyte stimulator, also called B-cell activating factor, and we've discussed this many times. And Lenny is always in the group and says, why does there have to be two names? It's so confusing. I really hate it. What's the real name? And so obviously the real name is TNF-SF13B, TNF superfamily, so that's not easy to say, so we'll just regress to the acronym. And so this is a B-cell survival factor. Peter described it in great detail. There is a closely related molecule called a proliferation-associated ligand. These are such cute names in a really subtle way, but they are necessary for the survival of B cells at different stages of differentiation. And why do we care about them as possible targets for therapy? Well, if we're just kind of quantitative biologists, because in some diseases there's more of them. So lupus, the levels can be very heterogeneous, but some patients can have very significant elevations. Rheumatoid arthritis, at least looking at blood levels, not as significantly and consistently elevated, but many patients are abnormally high, but there's actually very high representation at the site of disease. So what the heck? If it's too high, we should knock it out. And Peter already described these agents, and Belimumab was developed to be able to have an antibody that bound to soluble BAFF or BLYS and actually have no effect on APRIL. Other agents have different properties which are illustrated here, and you have the thumb drive. We don't have a lot of time to talk about it, but there can be complex interactions. There are 3 receptors. The molecules work as trimers, and there can be heterotrimers. So the variations are potentially great, and I think that we actually know relatively little about the biology, although we're learning more. So this was meant to illustrate an early trial with, I can never say this right, I don't know, Tabalumab or Tabalumab, whatever. I hate these names. But Tab is very, a lot of people find it very popular. Yeah, it's refreshing, yes. Actually, Tony Fauci used to be addicted to Tab. It was great. And then when it wasn't really in common production, he had cases made. It's just that saccharin. It's just, ooh. But this study showed at least clinical benefits in rheumatoid arthritis patients. This is just an ACR20 score, and many people were troubled that there was no dose response. But that doesn't stop people from moving forward in rheumatoid arthritis. Unfortunately, Belimumab actually was found to not have very great clinical benefits, but this is obviously an antibody that's new and improved because it binds BLYS in the soluble form as well as on a membrane of the cell. Whatever that does, nobody knows, but it must be a good idea. But one of the things that's remarkable is, just Peter pointed this out, it is remarkable and distinct that another way to target B cells actually increases the blood level of memory B cells, as opposed to what I showed you with regard to anti-CD20, which actually decreases them all. So we don't really know what this means, as Peter pointed out. It could be that it makes the memory B cells really uncomfortable in the tissues, and they run out looking for a safe haven. But one way or another, it means the biology is very different. We have to learn more about it, and the levels of B cell depletion is actually not as profound. But I just wanted to be reminded that, in fact, it was shown to have some efficacy in lupus in patients with non-renal disease. And there's about a 15% improvement in the SLEDAI score and in this combined responder index, and it has subsequently become the first agent approved by the FDA for the treatment of lupus based on randomized controlled trials, which is no small feat. So the delta may not be that great. Some people criticize it, but they found a path to success. So it makes us optimistic for the future. And I'll just point out that, in fact, Tabalumab does the same thing where it actually increases CD27 positive memory cells. So this seems to be a class effect. No matter how you block BAFF and APRIL, that's what happens. Unfortunately, recent announcement on the web said the phase 3 trials for RA for this agent were discontinued. They did an interim, what they call futility analysis, and they found that, yes, it was futile. So they stopped it. So there's something between ironic and disappointing in that. And it actually goes along that it seems to have similar efficacy as other trials that have examined BAFF blockers. And my question is this because of the odd effects on subsets of B cells? So if we switch back to anti-CD20, Rituximab was so successful, but it's so old school, we should be able to do better than that now. We are fantastic antibody engineers. We can make anything. Before we made a silk purse out of a sow's ear, starting with a mouse antibody. We made it, just glued it together. We can fully humanize antibodies. We can actually change the affinity. We can change their effector functions. Now, I'm going to talk briefly about the second generation agents, and one of them has actually been approved for the treatment of chronic lymphocytic leukemia, and third generation agents, which are even more exciting, what you can do. But a segue, we'll learn a lesson from a second generation agent. Question is, how do we treat patients with anti-CD20, and what is the regimen based on? And I understand, again, this is sort of my projection, that when they had the pivotal trial in non-Hodgkin's lymphoma, they decided what the minimum difference is between the control group and the treatment group for efficacy, and then they calculated power analysis, which we all do very seriously in our applications, how many patients they needed, and then they basically found out how much they could make in time and divided it by that number. So the dose, more or less, was very rationalized and empiric and not based on sequential studies and dose ranging. This is the only study that I know of that's actually even questioned whether or not splitting the dose of an anti-CD20 agent is better than just giving it all in one squirt. And I have to say, the way that I think about it is, if I drive my car, should I fill up the tank once, or should I fill it up halfway and then fill it up again? It's really annoying. So here they gave one big dose of 400 milligrams, or they split it in two. Okay, and there are implications, I have to say, between blood levels and how long they'd stay above a certain threshold, but does it matter clinically? This actually showed that if you split the dose, you had a statistically significant improvement in clinical response. And that was true at least for ACR 20 and 50. And it actually raises the question, what are our regimens based on? And they're based on legacy, I guess. And so, in fact, anti-CD20 might be, outcomes might be improved if we actually reconsidered, I would say, how we're treating patients. Ofatumumab is an antibody that's a second generation, which was designed to have greater complement binding activity. And in fact, as I suggested, that was one of the 3 or 4 possible mechanisms by which B cells are killed. And they actually fulfilled the prophecy that if you activate more complement with an antibody that you infuse at high levels, you will get infusion reactions, and you'll get lots of them. But in fact, it turned out peri-infusional steroids blocked that effect. These clinical benefits were really not significantly different from Rituximab, despite the capacity to induce more infusion reactions. So the Ocrelizimab agent, which I was referring to earlier, was meant to be the agent that would replace Rituximab. And in phase 3 trials, they had some unusual and unexpected infections, atypical mycobacterial infections, in particular in Asian populations. And it sort of sunk the boat, especially because they didn't see any difference in efficacy with regard to Rituximab. Let me just point out they were not PML infections. These were relatively small clinical trials. But the development of Ocrelizimab has been discontinued in rheumatoid arthritis, but in fact continues in MS. And Ofatumumab is soldiering on in both diseases at the present time. So let's get to the third generation already. What is the future? And this was an antibody that was developed in San Diego at what's called now Applied Molecular Evolution, but it actually doesn't exist because it was bought by Lilly. So Lilly wanted to get into the protein era. And this antibody, when it was made within a company that was led by Bill Hughes, they improved the antibody so it bound better to CD20 at higher affinity. They, up to 20 times better. They improved the antibody-dependent cytotoxicity, the ADCC, so interacting with Fc gamma receptors. And they had looked at it in a number of conditions, and oddly enough, they licensed it out to oncologists because they couldn't figure out in-house whether or not it would be better in RA. And this is a blurry picture, but it goes to show you that actually at 2 log orders lower doses depletes B cells as efficiently as Rituximab. This is called Ocaratuzumab, okay, but I'll call it OCAR. I don't like these names. And these are other studies that looked specifically at ADCC and showed, again, compared to Rituximab over a dose, these are test tube experiments, much greater efficiency of B-cell killing in vitro, even in individuals that have inherited the allele of an Fc gamma receptor that is the lowest affinity binding to Fc. So this is actually a bugaboo in non-Hodgkin's lymphoma. These patients don't respond very well when they had bad inheritance for binding through their Fc gamma receptor. But this antibody was engineered to get past that. But this was amazing to me because I actually think about stoichiometry all the time. I don't know why, but here they use 5 milligrams. You know, you use 2 grams, 1 gram twice with Rituximab to deplete B cells. Here they only gave 5 milligrams, and they had very significant depletion. The B cells started to come back, but you'd say maybe it's an artifact, maybe it's not real. Well, if you deplete enough blood B cells, your BAFF levels go up, and that's actually what they found. So this puny dose actually has great pharmacodynamic activity, and the question is whether it'll provide new opportunities clinically in our diseases, and we'll see. So this just compares these different agents, the old school standby, the chimeric antibody, part mouse, part human. It's some kind of should be in a Greek myth or something, but it has, comparatively, it has so much antibody-dependent cytotoxicity. Ofatumumab, they actually tried to lessen that effect. And OCAR, they thought this would be the way to go, and it may well have to do with its incredible B-cell depleting activity, complement-dependent cytotoxicity, strengthen Ofatumumab, and you get more infusion reactions, decreased in OCAR. And so here we have different approaches, and it may be that you can find a new way to skin a cat with a better agent. So the evolution, how these things have developed. I've actually studied B-cell toxins that target B cells and are very good at lowering host defenses so that some infections can win the game. IDEC actually made anti-idiotypes that were effective at killing non-Hodgkin's lymphoma. Rituxan was a new era. We're into the 2nd and 3rd generation. We have BAFF and APRIL targeting. And I have to say there's really fantastic opportunities if you make bispecific antibodies and you have the same antibody target more than one site. So let me just move on to just another topic, which is in rheumatoid arthritis, which we're focusing in on. The role of B cells, I would think, would be obvious because you have autoantibodies. And so looking at one of these characteristic antibodies, an ACPA, an anti-citrullonated protein antibody, one that's used in the clinic, CCP3, it must be good because it's 3. It's not the first one. It must be better than the second. Actually, they're all about equivalent. There's no difference in titers if you have early stage disease in the first 2 years, whether you've had it more and more years. But there are actually people that appear to have positive anti-CCP antibodies, but we're not sure what this means. Now, there are retrospective studies from blood banks that have suggested that these class of autoantibodies can develop 5, 10, and even 15 years before the onset of disease, but it's not clear that this is definitely going to develop into rheumatoid arthritis, so it's kind of puzzling what this antibody means. And it could be, in fact, that it isn't just the CCP3, which is a synthetic and unnatural antibody used diagnostically. People, in fact, with rheumatoid arthritis have autoantibodies to citrullinated versions of a number of different proteins. So it may be the specificity of the repertoire that's important. And so, in fact, what we found is that although there is a certain number of people that are first-degree relatives that have anti-CCP3 antibodies, They are a bit different than people with bona fide rheumatoid arthritis, because people with bona fide arthritis actually have a broader repertoire and are seeing different specific epitopes, not just the citrulline, but the protein itself. So there's an expansion and a repertoire spreading. But I have to say that the disease is not just antibodies, maybe. It's about recirculating B cells, trafficking in and out of joints, and whether or not B cells are actually, in a way, infectious for this disease. And maybe the people that have anti-CCP antibodies but don't have rheumatoid arthritis have not had it settled in their joints. That's what the people in Amsterdam would suggest. So I'm trying to figure this out, to tell you the truth. So we're actually comparing serum levels of autoantibodies to those made by blood memory B cells. And the question is there any relationship? Because you can, in fact, argue that the autoantibodies in your circulation are the products of long-lived plasma cells that are in your bone marrow. They were triggered decades ago, just like your anti-tetanus toxoid, and they really have nothing to do with the disease at all. Are there really recirculating B cells that are disease-specific? Well, here's your serum level for each individual, and then we took the memory B cells from the circulation, and we cultured them in vitro, we stimulated them, and we found out that they're directly proportional. So in fact, you do have recirculating disease-specific factors, and these B cells actually don't make antibodies to microbial antigens. And I have to say that it was such a fun test that we did it with about 12 or 15 paired antigens. This is CCP3, where there's a citrulline peptide. This is the reactivity for CQP, where there's only one amino acid difference. So it's actually a very fun assay. And the other thing that makes it super fun, it's a bead-based assay. So we have about 30 or 40 beads, each with a different antigen, so that we can actually test all these antibodies all at once with about 5 microliters. Actually, I'm lying. It's 1 to 1,000. So it's actually less than 1 microliter of blood. And we can do it with a little supernatant from tissue culture. So this makes me think that we can start tracking the impact of disease-specific memory B cells in rheumatoid arthritis and actually understand how these therapies may, in fact, be affecting the cells disease-specific from the adaptive immune system. So I have one last segue, which I think is super fun, because I might argue that the antibodies don't matter. It's all about the B cells. So here's a paper that's so recent it hasn't been published. It's just, this is the galleys. So they turn the paradigm around. We know that rheumatoid arthritis, if you get a diagnosis, your life expectancy is reduced by about 10 years. And it's mostly from heart disease. So they said, well, what about people that don't have rheumatoid arthritis? Every now and then you find somebody with an anti-CCP. What does that mean? So they actually looked at a collection of 3,000 healthy males, none with rheumatoid arthritis. It was retrospective. And they actually followed them over 15 years, looked at their blood afterward. And they saw who had a heart attack, who had angina, who was hospitalized for stents. And so they got a number of events, and that was 152. And so these people, they have cardiovascular disease. 10% of them actually had anti-CCP antibodies, and the remainder of the population, actually it was 3.8%. The risk of having a heart attack or stroke in this retrospective correlative study was greater than threefold if you had an anti-CCP antibody. So this is controlling for other factors, and smoking, and probably eating at McDonald's, I'm not sure, but CRP levels and other things. These people didn't have RA, but this suggests that, in fact, anti-CCP is bad for you, and that the cardiovascular risk, which has been reported to be only associated with seropositive patients, may actually just have to do with the autoantibody. So this is my concluding slide. I covered a lot of ground, maybe too much, I apologize. But anti-CD20 therapy is the paradigm. There may be new and improved ways of developing it in the future. We may be able to even develop dosing regimens that have better outcomes. There are other approaches that are being explored. I think one of the opportunities is to better understand what it does to the disease. And I think memory B cells is a topic that has not been explored, but provides opportunities for optimizing therapy for the future. Thank you.